want to do is introduce Jerry. He's a member of our group and has done other presentations for us. Today, Jerry, you're presenting on scientific dishonesty?
Scientific misconduct.
Misconduct.
So the topic du jour is scientific misconduct. And what we're going to talk about is three elements: fabrication, falsification, and plagiarism. But we're going to make this like a medical school. We're going to have a lecture and then have a live laboratory session to fabricate some information. So fabrication is a lie told when somebody submits a statement as truth without knowing for certain whether or not it's actually true. Although the statement may be possible or plausible, it's not based on fact. Rather, it's made up or it's a misrepresentation of the truth. Now, here's an example of, now here's one: falsification. Falsification is the manipulation of, in our case, research data with the intention of giving false information. It could be manipulation of images, which we're going to do, removing outliers or inconvenient results. And here's an image of Venice with ice, which of course can never occur. And this ice, it comes from another image and it's superimposed or it's integrated into this image. And then we have plagiarism, which is the practice of taking somebody else's work or ideas and passing them off as one's own. And by the way, if you use many other people's ideas, that's called research. So it's a little bit different. So the first one I want to show as an example is the saga of Diederik Stapel. He was the dean of the School of Social and Behavioral Sciences in Tilburg University in the Netherlands. And he presented a simple experiment. He showed the relationship between racism and untidiness, which I found interesting because I've been through the Netherlands. There's nothing untidy in the Netherlands. But he ostensibly went to a train station in Utrecht, and he did this simple experiment. At the train platform at the Utrecht train station, he had white volunteers fill out a bogus questionnaire just to keep them busy. And the white volunteers were invited to sit in a horizontal row of six chairs. The chairs were empty, except for the first one in the row. And seated in the first chair of the row was either a black or a white occupant. Now, when he fabricated data which reflected cultural expectations, and this is an important point, to satisfy expectations whenever you're fabricating data. And what he found was in his fabricated data was that white volunteers tended to sit farther away from the black person when the surrounding area was strewn with garbage. This was not true when a white person sat in the first chair, and it was not true when there was no garbage. And so his conclusion was that racism becomes expressed in an unpleasant situation, which is exactly what a Dutch intellectual audience would expect. So they would welcome this kind of result. But as it came out, 2 graduate students suspected some fraud. Somehow they went to the Utrecht train station and they found that the chairs at the train station didn't match the experiment. They just weren't set up to seat people the way it was said that Stapel had said that they sat. And they brought their concerns to Marcel Zellenberg. He was the boss, the head of the social psychology department at Tilburg. And after some prolonged confrontation, Stapel eventually came clean. Now, there were prior suspicions. In one study, Stapel discussed the statistical probability of the findings with a colleague. Okay, fine. And then at a later date, the same colleague asked to see the primary data, but was told it had not been yet entered into the computer, which of course would be impossible. He couldn't have discussed the statistical data that he hadn't entered into the computer. And there were other suspicions. Several of Stapel's grad students noted that they were included in study design and data assessment, but they did not administer the test itself. Stapel always seemed to have a friend at a distant school who would supply the subjects and help administer the psychological test. And the other suspicions, the main one was that Stapel's studies all seem to work out. That never happens in the real world. And in the investigation, they looked at why suspicions were not forwarded to academic superiors. And it was out of respect for Stapel's position and the students' fear for their own careers. And when Stapel finally came clean, he said, here's what he said: the journals wanted neat, clean studies with clear results. True. Clear, simple studies appealed to readers. True again. Getting grants had become a business which depended upon publications. True again. And he wanted to provide his student co-authors with good studies to advance their careers. What was interesting was that his own explanation, there was no acknowledgement at all of his own dishonesty or possible sociopathic personality. He just didn't get it. So he had all these external reasons why he had to do it. So that was just an interesting sort of brouhaha about fabricating of data. That's how people do it, or do it improperly.
What's the date?
I was in the 70s, I think. But we'll want to go back a little bit further in time to Gregor Johann Mendel. He was a giant in genetics, He defined basically what genetics was. And he lived at a time when the gene and the chromosome were not known. Now, the German animal and dye industry was going, for dyeing clothes, and they provided dyes for microscope specimens. So microscopes were pretty good then. So it was known there was a nucleus, and it was very chromatic. It stained with lots of things. So it was called chromatin, but it wasn't known that contained any genetic material. Now, Mendel had a hypothesis. This was the essence of his brilliance. He assumed that traits, at least simple ones, were transmitted as unitary elements, one from the male and one for the female. Perhaps to him it was God's plan, but this was just so fundamental to what was all about. And if you express it mathematically, this kind of looks like a quadratic equation, which it's not. Now, I used a capital A just to show a dominant, and I use a small letter to show a recessive. So if you have a heterozygote, meaning a dominant recessive, mated with another heterozygote, overall, you're going to expect one homozygote dominant, AA, one homozygous recessive, AA, and two heterozygotes, big A, small a, big A, small a. And that was, he expressed it this way. And his assumption was that inherited traits, at least simple ones, were transmitted as unitary elements. So you have pollen, you have the egg and the silk, and you have you have the pollen here, one part of a corn plant, and you have the egg, another part of the corn plant, and the bees or whoever, or the wind carries these around. And the modern view, just to give us our own terminology, we have a male gamete, which is a haploid cell with a single set of chromosomes. We have 23, but we actually have 23 pairs of chromosomes, whereas the gamete has 23 chromosomes, and that fuses with the female haploid gamete, and that's the essence of all reproduction. Or not all. Now, we've known this intuitively, that children look like their parents. Here is Prince Harry. And here is Captain James Hewitt, who you might remember was Princess Diana's boyfriend. And the resemblance is, of course, uncanny. What this gentleman is doing here, this unhappy man is doing here, is not clear, but certainly time has shown that some of the relationship between this man and this man may be related to this man. If there was any question about it, we certainly have the ability to settle the issue, but that's for another talk. Now, Mendel's bean plants were an excellent model, just a great model. They had 7 traits with a dominant and recessive gene. He used the peas and the pods mostly. And plants could self-pollinate. So what you have peas, you have a green pea, smooth or wrinkled, and we're going to use this one, green pea, green or yellow. And it had pods. Now, yellow is the dominant. The pod and green pods would only contain green seeds, and yellow pods would only contain yellow seeds. So he knew what was inside the pod just by looking at it. But there were other single gene traits, purple versus white. Some were tall versus dwarf. Some had an axial branching where all the branches took off at one point, and another one where they grew on the terminal end. But we're going to concentrate on this one. So hybrid plants for pea and pod color had both yellow peas in yellow pods and green peas in green pods. So looking at the genotype, to review this, Mendel's plants, because I know this is not your field of endeavor, Mendel's plants produce all yellow pods with only yellow seeds, green pods with green seeds. But some plants produce both yellow pods containing yellow seeds and green pods containing green seeds. Now that's a little bit idealized, as anybody who's grown plants would know, is that not all blossoms flourish and not all will come out. So it's possible that a given plant could try to produce both yellow pods and yellow seeds and green pods and green seeds, but it could fail in some of them, and just some of them would fail. And you could have all yellow pods, or you could have all green pods. But he surmised that hybrid plants, YG, produce both yellow pods with yellow seeds and green pods with green seeds, but some would fail. And here's what he expected. He took a yellow plant and he mated it with a green plant. This was all yellow. And he was hoping that this would be YY, two big yellows, and this would be GG, two greens. And he would get the first generation would have one yellow trait and one green trait. He didn't know about genes. One yellow trait, one green trait. And so would the second one, so would the third, so would the fourth. And if he took this one alone and self-pollinated, he would ideally get one dominant YY, two recessive YG, two heterozygotes YG, and one GG. And then he would select this one out and self-pollinate, and he would again expect this 3-to-1 ratio, 3 yellows, these would be mostly yellow, and one green. So here's what the ones that are YY are mostly yellow, they can have a green pod. So here's how it works. One the same as before. One dominant, one recessive, one dominant, one recessive, and you're going to get 3 mostly yellows and 1 green. But you can have some green seeds in there. And he could look at a particular plant. If it had yellow and green in it, he could then look at that and say it was a heterozygote and breed that one. And if he was trying to get a pure strain of YG, so he bred them for 10 generations. And so if he had a 75% chance of getting this combination and 25% chance of getting this, it would basically be 3-to-1. So that over 10 generations to getting a pure YG would be colonies would be 0.75 to the 10th, which would be about 1 in 18. So now here's a sample experiment which raises the red flag. He did a hybrid experiment and he found of 258 hybrid plants, he got 8,023 seeds. As yellow as the dominant color was 6,022. Green was 2,001. Expected ratio 3-to-1, and he got 3.009 to 1. That's so right on. And he did many experiments. He had assistants as an abbot in the rectory, and the data reported by Mendel, to say it briefly, was too high. And we'll go over this. Was too high a frequency, being too close to expectation. And now we get into the real math of it. Outcomes of experiments relying on random chance would be chromosomal segregation is independent. They have their own distribution. 68% of the experiments are expected to be within 1 standard deviation, and 32% would be outside 1 standard deviation. And now we get to the problem of the standard error. If you take a small sample from a large population, that small sample will likely deviate from the larger sample. And we can do that in an intuitive level. We're at Super Bowl 57. And if we look at the last 6 Super Bowls as a small sample, and we look at Puxatawny Phil, shown down here, Puxatawny Phil is the groundhog that they take out in Pennsylvania. And if he sees his shadow, there's 6 more weeks of winter. If he doesn't, there's something else. In the last 6 Super Bowls, when Puxatawny, including this one, which Kansas City won, in the last 6 Super Bowls, if Puxatawny Phil saw his shadow before the Super Bowl, the American Football Conference won. And when he didn't see his shadow, the National Football Conference won. 100% correlation for a small sample. Whereas if you look at all 57 Super Bowls, it's about 50-50. So that has to do with the standard error. So the range of deviation of small samples increases as large as their size decreases. Is that clear to everybody?
Yeah, that seems clear.
So when a scientist presents a bunch of small samples, here we go, small samples of random process, there should be some deviation, the standard error from the large population. And to make things worse, Mendel had studied advanced mathematics and done statistical work on sunspots and other celestial events. The standard error was well known at the time that he did his experiments, and it seems unlikely that he would not be aware of the standard error. And just a few other examples, he used wrinkled rounded seeds, 253 plants, 733 seeds. Round was 5474, wrinkled 1850, which is 3.38 to 1. Very close to the perfection there. And yellow versus green seeds, again, 6022, 2001 ratio 3.32. Very close to the expected ratio. So the probability here is of getting a perfect result would be 1 in 18 or so, 1 in 17. But Mendel's results fell in here within 1 standard deviation. Multiple tests from a random system should fall into the bell curve. The sum should have been outside of here. So Mendel bred himself the hybrid plants 10 times. With each generation, he bred only 1 plant, and his results were too good to be true. As we've said, too close to 3 to 1. And this was discovered in 1936 when Ronald Fisher, who was a famous statistician, he analyzed Mendel's results. Now he's British, so he has the typical British understatement. So he said Mendel's data came out too good to be true. So did Fisher spill the beans? Well, here's what he wrote in his typical British understatement. It is interesting that Mendel's original results all fall within the limits of probable error. If his experiments were repeated, it's the individual ones, the odds against getting such good results in any one test is about 16 to 1. Here's that number rather than 18. It may have been luck, or it may have been that the worthy German abbot, in his ignorance of probable error, unlikely, unconsciously placed doubtful plans on the side which favored his hypothesis. Or simply put, Mendel's data is too good to be true, and Fisher wrote, I get this from his article, a total deviation of the magnitude observed and in the right direction, this is for all of his experiments, he put them together, is only to be expected in 1 in 444 trials. There is therefore a serious discrepancy. And his mentor, W.R. Weldon, wrote, Fisher's mentor, because Fisher ran it by him, Mendel cooked his figures, but he was substantially right. It is far more likely that there was some manipulation of data to fit his hypothesis, i.e., more of a demonstration than primary research. So if Mendel did cook the books, but was essentially right in his theory, was this misconduct? It was. It was fabrication, a lie told when someone submits a statement as truth without knowing for certain whether or not it was actually true. Did Mendel get a pass from the scientific community? Yep, he was right. His theories were right, and they were actually fundamental to everything. He had no ulterior motive. He was an abbot. And when he was promoted after 8 years to another position, he just stopped doing research and never made any big deal of it. He wasn't going for academic promotion or anything. And his data satisfied Occam's razor. Sir William of Ockham was a religious figure around the time of Calvin. And his razor was that the simplest explanation is the best. And scientists love these very simple explanations when they turn out to be true. So he was given a commutation. He's given forgiveness. Now,
wasn't he also forgotten? I thought he had to be rediscovered later.
Yeah, he was. He was rediscovered. You're quite right, Charlie. He was rediscovered sometime after 1900, and that's what got Fisher to look at it then. You're quite right. He was forgotten, but science was moving on, so the data was especially important by that time. So now, if we're looking for scientific misconduct, it's kind of like the atmosphere. It's all around us. So let's try to find one ourselves, and we're not going to have to look too hard. And so I picked something to look at that you'd be familiar with. Now, when you look for, say, rotten corn, you look in a cornfield. You don't look in a wheat field. And if you're looking for untruth, you look in the Soviet Union, because there, truth is a very flexible concept, as we saw in Putin's speech. That was yesterday, the day before, his annual speech. Truth is a flexible concept. Now, the Sputnik V is the Russian COVID vaccine, and it does work, and it's a good product. I'm not saying it isn't. And this was published in The Lancet. They took, which is very well known for trying to be on the cutting edge, and sometimes they're on the bleeding edge. The Russians used a tried and true method. They used a DNA virus, attenuated DNA virus, adenovirus 26. Adenovirus 26 has been used for years to deliver DNA that takes over the cytoplasm and manufactures whatever protein you've added to their DNA. Whatever nucleic acid you've added to the DNA to make whatever protein or whatever substance you want to make. It's tried and true. The challenge for the researchers was that about, I think it's 15%, maybe 20% of Russians have had adenovirus 26, and so they have antibodies to it. So they didn't want to. They'd have people who wouldn't respond to the vaccine, they felt, and so they used a second, less common virus, attenuated virus, adenovirus 5, which virtually nobody in Russia has had. And one shot will do the trick. But to cover the people who've had adenovirus 26, they gave a second shot of adenovirus 5. Tried and true. And they gave a 21-day interval. This is the usual boilerplate stuff for the second dose. 19,866 received it. No serious adverse effects. Just what people would expect. And I'm sure it was true. But what they said was, whereas here, vaccine efficacy was 91.6% overall. That's an important number because the Russians wanted to show, I believe, this is my feeling, that their vaccine was better than the American British ones. So let's look at the one made by Pfizer and BioNTech. What they reported was that it was their vaccine in their trial was 100% effective for between age 12 and 15, 95% effective 16 and older, up to age 65 group was 94% effective, and then it dropped off in efficacy. There's a little variation here. It was less effective in people than 70 versus 80, but that's how sampling goes. But what they showed was there was a decline in efficacy as expected. There's a decline with age, and that's what you see in all viral immunization. And if you look at Moderna statistics, again, the same thing. 95%, 95.6 under age 65, 86.4 older than 65 in prevention of disease. Now, here's what the Lancet article, if I took this from the Lancet article, here's what the Sputnik V was reported to show. Overall 91.6, and look at this cluster, 91, 90, 91, 92, 92. That's a red flag. And so people said, well, let's look at the primary data. Now it's standard nowadays when people have articles published in paper journals like the New England Journal of Lancet, they publish the primary article and then the main article, and then their primary data is just published online. So anybody can get it, but they don't want to waste the paper, and it's expensive, which is certainly appropriate. So so Lancet inquired to the Russians about their primary data, and the Russians refused to post it. That's known as a red flag. It doesn't say the data isn't true. The Russians said that's the data, but it's a red flag. Now, I took this graphic out of there. I just want to show you something interesting. It's not quite labeled probably with this red line, but I'm going to show you. These are the levels of antibodies to the receptor binding domain for the M spike that's generated by Sputnik V. Now, these aren't absolute numbers. These are ranges. But if you look at this is especially odd to see this, where age 18 to 30, they all cluster right here with very little variation. And this should have been down, it should have encircled these two. They cluster with very little variation. And all the others have a lot of variation. They don't cluster the same way. That's very unusual, meaning almost unheard of. Now, the biological explanation is that the scatter of values, whether it's male, female, male, whatever it is, should be similar. The scatter should be similar or show a trend in age. Or if you want to break it down arithmetically, the arithmetic and the geometric means, is a geometric mean for those who know about it, should be similar in all classes and trend with age. That's what they just should do that. This is a red flag that they didn't do that. So what's missing? We have no details on their PCR testing. And now we don't know how many PCR cycles were done to find COVID RNA. And that didn't influence the positivity rate. If you increase the RNA multiplication cycles in the PCR, the number of false positives increase because you get background scatter. So you get increased positivity with increase the number of cycles. And if you want to decrease the number of true positives, you just decrease the number of cycles, and that will increase the protection rate. So when you don't know how many cycles they were doing to what people, it's just simply you can't figure out what they did unless you're me. Okay, something to hide. The authors of Sputnik V have still not released their primary data, so let's just look around. Okay, Sputnik V, which does work, was sold at a very reasonable price, $8 a dose, very reasonable. Might be close to cost. And it was sold to the countries, anybody wanted it, but it was sold to the countries under the influence of the Soviet Union, but also sold around the world, South America, Central America. So let's just look at what other people reported. Now, this is actually from Russia. A guy named Barchuk found the Sputnik V to be 58% effective. That's not close to 91. In Mexico, the Sputnik V was 78% effective. Well, now Moderna was 91. Down here in Serbia, which is close to the Soviet Union, the Sputnik V was nice and high. And in Hungary, where it was 86%, sort of neither here nor there, but it wasn't 91%, not even close. And they got 91% in every group. Now, is this a tempest in a teapot? Sure it is, because it's a good product. But some of the data on Sputnik V was probably fabricated just to look better than the American data. But the vaccine does actually work well overall. And the basic principle of using attenuated viral viral vectors to deliver DNA to make an antigen is standard technology. But it's just interesting to look at their presentation. Now, the last case I want to present is the tale of Vijay Soman. I love this one. Henry Kissinger said this: academic politics are so vicious because the stakes are so small. And that really says it all. Now I want to show you this nice little model for looking at insulin. It's a nice little area of expertise. You get to publish papers. Now, here's a red blood cell, and glucose will enter red blood cells. Shown here is the red blood cell with a glucose channel that's closed, and here's an insulin receptor. The insulin's out here, and the glucose can't get into that cell. Now, we're going to take this insulin, we're going to put it on the receptor, and lo and behold, the glucose channel opens. And sure enough, the glucose will find its way into the cell. And this is a very, very convenient model to study the behavior of insulin receptors. Blood is easy to get from animals and humans. And you don't have to worry about storage smart. It's easy to store. It's just a good scientific model to publish some interesting little papers. And what's known as insulin receptors behave differently in health and disease. People get sick, the blood sugars go up. So let's look at this. Now, the model that's a good model to look at is anorexia nervosa, because these people are in good health physically, if not mentally. And they have chronic starvation. And chronic starvation, as expected, results in low blood glucose and low insulin levels. And these people will have an elevated blood glucose with a glucose tolerance test when they give them a good slug of glucose to drink, even though there's a marked rise in insulin. So the insulin goes up, but the blood sugar doesn't fall. And interestingly, the abnormality resolves with the restoration of normal feeding. So chronic starvation affects insulin receptors in some manner. Okay, nice little research area. So let's get into it. Now we have the cast of characters. Here's NIH, here's Yale. Cast of characters. NIH, National Institute of Arthritis, Metabolism, and Digestive Diseases. Now the researcher is Helena Robard. I don't speak German, so I'm going to just call her Helena Robard. And she's the junior researcher doing the bench work. And Jesse Roth is the chief of the diabetes branch at that institution. And then Robard and Roth, two R's. At Yale School of Medicine, we have Vijay Soman. He's the junior researcher doing the bench work. And Philip Feelig, vice chairman of the Department of Medicine and the chief of endocrinology. Now, so Helena Robard, young researcher in diabetes, NIH 1978. That's her current image. Now, she submits a paper to the New England Journal of Medicine on insulin binding on red blood cells of a patient with anorexia nervosa. And Jesse Roth, her senior, is the co-author. Okay. So the paper goes to Bud Relman. He went to my medical school, so I have to say that, Columbia PNS 1946. But he's over at Shattuck Street where the New England Journal of Medicine is. Now, he's infectious disease. So he sends the article, which is standard practice, to 3 authorities, 3 reviewers, all specializing in this area. And one of the researchers gave it a negative review. And here we go. Philip Feelig at Yale gave the paper for review to his assistant, Vijay Soman, who was working on the identical subject in Felig's lab. Shortly after receiving the paper, Feelig sent a paper on the same subject with Vijay Soman as co-author to the American Journal of Medicine, not the New England, the American Journal of Medicine. So they know nothing about what's going on in the New England Journal of Medicine. And who do they send the paper to review it? Well, yeah, they sent it to Jesse Roth at NIH, the very same author of the original paper submitted to the New England Journal. And he appropriately passes it on to his junior, who does this kind of work, Helena Robard, for her review. So what we have Soman and Feelig reviewing Roth and Robard, and we have Roth and Robard reviewing Soman and Feelig. Alas, when Helena Robard at NIH reads the paper from Yale, as a quote, she was aghast. Here was her paper, complete with verbatim passages and even a formula she had devised. Ha ha. So now what happens? Feelig flies down to NIH and talks with Roth. Feelig convinces Roth that there's minor plagiarism, but the work was done before he received the NIH paper to review. This turned out to be untrue. Feelig agreed not to publish his own paper if Roth felt there was more than minor plagiarism and that he would withhold his publication in the American Journal until the Roth-Robard paper was published in the New England Journal of Medicine. Feelig did not keep his word. Helena Robard, this is where you see the stakes are so small. Helena Robard believed that the Yale paper was entirely fabricated and demanded a review, or she would, now get this, denounce Feelig and Soman at the upcoming Federation of Clinical Research meeting. Whoa, that's it now. And so Joseph Rall, Roth's boss at NIH, was chosen as the auditor to examine Soman's data. So NIH's Joseph Rall's audit, never done. He said he was too busy and it never got done. So by this time, now here we are, 1979, Soman and Feelig have published their article in the American Journal. And Feelig at Yale accepts the Bard Professorship and position as chairman of the Department of Medicine at Columbia PNS. I remember when this happened. Now, this is the Bard Fellowship, and this is the dorm Bard Hall. And if you see this red square, that window there is my window in room 407, where I lived at Columbia. I thought you'd all want to know that. And he starts in June 1980, and he brings Vijay Soman with him. Now, Yale springs into action, and that needs an asterisk because they're not worried since Feelig and Soman are now ensconced at Columbia. So they bring in Jeff Flyer, which some of you may have heard of. He's at Harvard. He's brought in as the reviewer. He's been at Harvard for years. I know his brother. Vijay Soman confessed to fudging and fabricating. He said that Feelig did not know about it. And Yale advised Soman that his best choice was to resign and give up research. A few weeks later, Soman at Columbia disappeared into India. But Helena Robard insists that another reviewer be brought in to review plagiarism, not fudging. Again, Yale Medical School springs into action, and they bring in someone from University of Colorado, Joe Olefsky. He examined 14 of Soman's publications dealing with insulin receptors. Only 2 articles had supporting data. Feelig had co-authored 10 of the other dozen that had no supporting data. So we have plagiarism and fabrication, a definite at this point. When the dust settled, Yale had retracted 12 papers by Feelig and Soman. Soman's vanished into India. Feelig started as chief of medicine at Columbia, June 1980, and resigned his position in August 1980. He was gone. And the epilogue for Philip Feelig is Yale rehired Feelig as a tenured full professor, but not to his original Dowd chairs. He was found not guilty of fraud by an NIH investigating committee, and his major NIH grant was renewed. He left Yale to work briefly for Sandoz, probably was pushed out, and he started a private practice in New York, whereas Helena Robard became president of the American College of Endocrinology, president of the American Association of Clinical Endocrinologists, master of the American College of Endocrinology, and went into private practice of endocrinology. Okay, so now we've done data fabrication. So what I'd like to do is, I don't know if I can make this a little, no. So I'm going to show you how to fabricate some data. I'm going to have a little user's guide here. Is everybody with me on this? Because I don't hear anybody. I can't hear anybody.
Yeah, we're still here.
Okay. Now, the first step in this whole process is to understand the correct process of data collection so that fabricated data will ring true. And George Lichtenberg, he was a German scientist and satirist. He wrote, and this is the essence of doing this stuff, is the most dangerous of all falsehoods is a slightly distorted truth. You got to keep this in mind of falsifying data. You only can distort the truth slightly. And what we're going to work on, and I'll explain how electrophoresis works later, we're going to use electrophoresis data, which can be used for identifying DNA, RNA proteins, find mutations. But the important point is the results are pictorial. And this gives credence to findings, unlike the Russians, which just gave numbers, which who knows what they gave.
I'd like to comment on that remark about slight distortion and bring up the question of the big lie and where does that come in?
In science, that doesn't fly. When you say, I've measured the hematocrit of green men from Mars, it doesn't fly. That would be too quickly show that how you got there doesn't work. The big lies don't work in medicine. There's too much data to falsify, to show they're false. Big lies are effective in propaganda, where you keep saying the same thing over and over and over as a lie, and eventually becomes true. That doesn't work in medicine. It just doesn't work.
This is restricted to this area.
Oh yeah, we're talking about scientific misconduct. That's what we're doing. We're working on scientific. We're scientists here.
Thank you.
Okay. But pictorial results are good. So here's our slightly revolutionary experiment that we're going to make. Yeah.
Now, just a quick point. You're using the term science and medicine interchangeably.
Yeah.
And there's plenty of evidence in certain areas of science where lots of money is at stake, that little cliques, self-referential bunches of people can create a big lie. The classic example, of course, is the hockey stick in the so-called climate research that was perpetrated by Michael Mann and then the
I think they cooked their data. I think they cooked their data, didn't they?
Yeah, that was cooked data.
Yeah, and now they have a worldwide audience and who wanted to hear. But you're right that they did have the big lie, but they also adhere to a basic principle of finding the expectations that the audience wanted to hear.
Well, that's true, and it also fed into the idea that there was a lot at stake in the sense of many millions of dollars in funding.
They want right, exactly, as Staple showed. Yeah, they were right on the mark with their falsification. It just didn't work. So now what we're going to do is we're going to take a strand of DNA from a fruit fly, that's Drosophila, and we're going to insert it into a human bone marrow stem cell. Now the stem cells, they're called bone marrow stem cells. They actually circulate they're actually in the circulation, and they look to the eye under the microscope like lymphocytes, but the surface markers are different. They can be identified. And that DNA produces a protein. We'll say that it enhances immunity in offspring lymphocytes. So that might be something of value to human beings if you could enhance their immunity using the Drosophila as the source. So it sounds appealing, just like the hockey stick sounds appealing. So our goal is to show that the DNA strand that we incorporated into the human bone marrow stem cell is replicated and subsequently found in lymphocytes descended from them. This is a good idea, great idea, but it is a big job, and there's no sense in wasting time actually doing it. Now, to create data, you do have to know something about it. Scientific experiments and methods, real scientific data, image manipulation. This is a craft. So we need to create a demonstration using gel electrophoresis of DNA from lymphocytes descended from our genetically altered bone marrow stem cells. And what we're going to use is electrophoresis. This is the overarching term. It's the movement of charged particles in a fluid or gel under the influence of an electric field. Now this breaks down into cataphoresis, which you move positively charged particles, cations, and anaphoresis, which we move negatively charged particles, which are anions. Now, almost all things, proteins, DNA in medicine are negatively charged. We never use those terms. We only say electrophoresis. So we're going to do a practical explanation. I think I'm going to go to my, just show you electrophoresis now, and then break from this and then go back to this. I'm going to, okay. I'm going to minimize this. Well, I'm just going to bring up my paint program. And I just want to show you what electrophoresis is. What we're going to have is we're going to have a plate. You start with a plate, and I'll give it some lines to give you a little perspective. We have a plate. And what we're going to pour into the plate is agarose. And you don't know what agar is. Agar, you take seaweed and you get the pectin out of it and the proteinaceous stuff and other stuff, and you're left with agar. And at least in my day, agar came in these brown glass jars as a powder, and they just add some water to it and it sets up as a gel, and it forms a lattice like this. And then it comes with a tool, and the tool is just a little plastic thing. It's like this. And the tool has whatever many teeth you want. I'll just give it 3 teeth. And you take this tool and you press it into here, and you get wells. And the wells, so the specimen can't see the surface. It goes into the well. And then you can take whatever your specimen is, and this is going to take DNA. Now, DNA is an acid, deoxyribonucleic acid. And so the conjugate base would be deoxyribonucleose, and the hydrogen ion come off. So we're going to take some electricity, and we're going to make this. We wanted to drive it away. So we're going to put electrons over here, and this end will be the positive end. Now, when we prepare some DNA, you break it up into fragments, and you pour some in the well. We'll pour it into this well, and you turn on the juice, and you go away, and you come back the next day, and what you'll see is these lines that have formed, deposits have formed. Actually, you don't see them, but they've formed. You can see maybe see them a little bit. But then you take a stain like a thinamide, a DNA stain. You stain it, and you can really see them. And what's happened is that the very small DNA fragments can travel this far through the lattice. They can just get through. The medium size go this far, and the big ones go there. So you can separate different DNA, so we know they're different DNA specimens. Okay, so now we know how gel electrophoresis works. Oh, okay. So now we're going to talk about image manipulation. There's 3 basic things in an image. It's its brightness, its contrast, and its gamma, which is nonlinear contrast. And gamma is the relationship between the numerical value of a pixel in an image file and the brightness of that pixel when viewed on screen. And its relationship is nonlinear. When you increase the voltage, the brightness increases by this factor on most monitors, which is the gamma. Now, that means a very high gamma results in rapid changes in brightness, which is useful for separating 2 areas close in color. An extremely high gamma between 2 areas close in color will create a sharp border between them. But more importantly for our fabrication, we look at low gammas. We want to create smooth transitions. They look realistic. Now, all of us have probably adjusted the gamma of our monitor screens in their entirety. In our graphics programs, we're going to use CritUp, which Bob introduced me to, Steve, one or the other. They permit changing the gamma of a particular brush stroke or line to give it a sharp or indistinct edge, which is important in fabrication. So here's a window with a gamma adjustment. Here we have an entire field, and it goes from white, snow white to blizzard white to jet black. But we want to look at this area here. So we want to increase the gamma here. And if we do something like that, if we look this area, this is going from slightly light to slightly dark. If we increase the gamma right here, we can look at this small area here and see the distinction. And we use this in reading CAT scans all the time. You just have a cursor. And if you have 2 things in the lung that are fairly close, but you want to see them better, you just move the cursor around and you play with the gamma. Now, the image manipulation trick is a partially imposed semi-transparent lines can create the illusion of a low gamma, add seamlessly to a true image. Okay. Now, I want to show something which I talked about in another talk. Our friend Sapan Desai, he was a Ganef. For those few of you who don't speak Yiddish, I'll tell you what a Ganef is. Carl knows what it is. A Ganef is a thief, But it's a thief. It's not a person who steals milk to feed his child. A Gandalf is an inherently dishonest person, would rather have a dishonest quarter in their pocket than an honest dollar. And everything is dishonest about a Gandalf. But the problems for Gandalfs is they always leave a trail. So Sapan Desai submitted a falsified paper on chloroquine hydroxychloroquine to the Lancet. You may remember this. It's a big brouhaha. And he had this company that had was ostensibly providing information services to all these hospitals in Africa, 250 hospitals. None of it was true. And he fabricated an article to the Lancet, and it was eventually discovered and disgraced, etc. But the point is that he leaves a trail, not that he got caught, because he was stupid what he did. So I look back at his PhD thesis. Now he was given some crazy stupid project about the inner ear of rodents and their neurons. Now that's from his professor. He didn't pick the project. We don't dispute what it was a silly project, even though it was. He presented some of the bony findings with neurons, but if you look, and it proved some point, but if you look at this graphic here, if you put a circle here, these are the same in the bones. These are replicated. This never happens. These two here with one sideways, never happens in nature. That never happens. And likewise, this whitish arch here, identical. That never happens in nature. And if we do go down to the pixel level, which is more the point, you can take this set of pixels and this set of pixels, and they are exactly the same. That could never happen in nature. So we know he falsified his data even as a grad student. And again, looking at another area of whiteness, he falsified that. He did a simple cut and paste. And so he was easy to catch. He left a trail all the way. So now I want to invite you into my laboratory so we can do some experiments. We're going to do this live, so we're going to see if this works. Now, here I've got a gel electrophoresis here, an old picture sitting around the lab. We're not doing any work here. Now, the two lines in the middle of the first band in column 2, right here, are pasted into column 3. This is just a cut and paste, nothing more than that. Now, the reader expects in our experiment for the new Drosophila gene to be very similar, but not exactly the same as column 2. So it's going to have this stuff, kinda, but we're going to put a new gene in here. But we'll get caught in a second with this. If you look carefully here, see that groove right in that line there? Can everybody see that groove? Can everybody see that groove?
Yes,
You'll get caught in a second there.
some do, some don't.
Well, there's a whitish groove. There's a whitish groove here, and then you got this downslope here. You're going to see these pixels are the same. See these pixels here, this pixel there. You're going to get caught in a second by just cutting and pasting. We know that. Okay. So what we're going to do now is want to use our graphics program. Now let me just get my snipping program up. Now I'm going to take this. Now we're going live here. I'm going to take this graphic, and I can leave a little border in here because we're going to get rid of the border later on. Okay, now that's in memory, so I'm going to bring up Krita. Krita is a graphics program like Photoshop, and either Steve or Bob showed it to me because it's free and it has a very steep learning curve. But there's a million videos on YouTube to learn how to use it if you're so inclined. And what we want to do is we want to start a new file. Okay, now I'll go quick, but I'll show you what I'm doing. This is a new file, and it starts with a background layer, which nobody ever uses it. You can have it have some squares used for alignment, but nobody ever uses it. And it's locked just because we don't want to fool around with it. Now we have paint layer 1 here, and paint layer 1 is in front of the background. But we're going to need more than one layer, so I'm going to go to this square here. I'm going to put more layers in. Now, the way this works is like the old Disney celluloid, where paint layer 1 is beneath layer 2, which is beneath layer 3, which is beneath layer 4. So if the cartoonist paints Mickey Mouse's body on layer 1, he can put Mickey Mouse's arms in layer 2, and then he wants to move them. He can make layer 2 invisible or erase it and paint layer 3. And that will always be in front of layer 1, but he won't erase part of layer 1 and have to redo it. So we do layers, and these layers in the graphics program can be either transparent or opaque, depending on what you want to do. And if you want to work on the layer and you don't want to ever touch it again because you just don't want to touch it because you need it, you lock it like that. So what we're going to do is we're going to drop our graphic into the lowest layer. It comes out as a layer 5. I don't know why it doesn't go into layer 1, but I'm going to move it down to make it layer 1. And then I'm going to, if I wanted to make it disappear, I go like that. And I'm going to lock it because I never want to touch that. And now we'll go up to the layer on top of it. Now we're a little close in to do graphics work, so let's just magnify it. Okay, this was the white line I was referring to that we'll get caught on. Okay, so now this has brushes, and there's many brushes here. And what we're going to do is we're going to pick a brush to draw some straight lines, and I'll tell you why. This is just a straight line brush. Now, the brushes have, brushes have many qualities that you can show. This one has only a few qualities. I can show you another one. But what we're going to do is we're going to draw some horizontal lines for the eye, because it's hard to make a good horizontal line on the fly. It's better to have a reference line. So here's our color gray here, up here. I'm going to change it to red. This gives you a choice to have 2 colors. I'm going to make a horizontal line. And let's make here, you've got to just do a marker for me. I'm going to use my graphics pen. I've got a graphics tablet, $65. Okay, so I'm going to make a line, and I need the line to be thin. Why am I not getting this? What have I done? Oh, see if this appears. Why this is not appearing? Why is the graphics down? Oh, now it appears. Oops. I could erase this if I feel like it. Let me see if this will. Why is my graphics tablet not working? The one thing I didn't check this morning. Oh. Why is this not working? This works, but my cursor works, but my graphics tablet won't work. I'll have to work with my cursor. Not as easy. We want to just create a thin line, and we want it horizontal just as a marker. So I'm going to get rid of all the jaggies so it'll be horizontal. Oops, I have it on erase. I'm sorry. Oh, that's why it wasn't showing. Oh, doesn't work. Close enough. Now we're going to have this line here. This will be our marker. Now let's say we'll put our Drosophila here, and I need something in this well here. Doesn't look right. So, yeah. Now I'm going to go to find another brush with a low gamma. This one here, it's got a fuzzy edge to it. Now I'm going to, this is a paint, we're on layer 1, so I'm going to lock this layer so I don't fool around with it. I'm going to layer 2. So now I'm on top of it. Now I want gray, so there's many ways to get colors. You have a color wheel that you can go to. But we're going to stick in the gray, so it doesn't matter really where this color wheel is, because we're just going to be dealing with grays. So I'm going to take this, I'm going to decrease its opacity because I don't want it to be too dark. And then I'm going to increase the size. to about this big. So what's that? What is 24 pixels? So let's draw a little something. So it's got to decrease the opacity. Now what I wanted is a little bit irregular. So the first one I drew horizontal. Then I want to take some irregular stuff. Okay. Now I have to blur the edge for the gamma effect. So I'm going to draw a line here. Okay, you see it's getting a little bit fuzzy. And then I'm going to fuzz it more by taking my brush and go dot, dot, dot. No, it's too dark. Let's make it a little lighter. I can go over here and make the gray a little bit lighter. Yeah. Oh, I see why. Its opacity is a little bit too much. I want to get its opacity down here. See, now it's starting to gray out. Can everybody see how we're getting the gamma good there?
I mean,
So-
you're a very skilled fabricator.
Hey, this is important stuff. Okay, now we're going to increase the opacity a bit. And now I need this bigger. This looks like it must be 90. Oops. So let's make the size 90, 80. Too big. That might be just a bit too big. Let's go closer to 50. That looks better. Okay. So again. Okay, now we're going to just keep repeating ourselves. Fill it in, a little angle here, a little regularity here. We don't want it too straight. I want a little central density. See again there. Put down just a little bit. Out a little bit. Okay. Now here's the trick. See this down slope here? We gotta create this down slope and this upslope in the gamma to make it look real. So let's find another brush with even less density. Now, oops, now it's so big. Let's make it, we're at around 50 or so. And let's get its opacity way down. So leave a little room for error. No, we need it much wider. This looks 90. Okay, now I can go below. I can go down below because we're going to cut this out. I need a little more width there. You know what? I want to go to another layer. This is not looking good. See, it's erasing. So let me go up to another layer so it won't erase what's underneath it. I need a little more opacity. It's not going good. It's too low. It's too light here. See, it permits you to go back. See if this is dark enough. Nope. I'm going to go with that and then darken up the. Okay, now I'm going to have to darken up the this line. So let's get a little, go to our color wheel. Let's get a little darker gray. And we need about, what was about 50? Pass these 100. Let's make a little bit. Not getting a dark enough color. Let me see what I'm doing here. A little angle on that to make it a little rougher. Okay, now let's reduce the opacity, just do a few dots. We gotta Oops. I have to soften this. We need that gamma to look right. Soften this, get this gamma to look right. Now let's just see. Maybe that's a big bag. Let's see what it looks like. Not too bad, not too good, not too bad. But I don't have somehow my graphics pen doesn't want to work. I don't know why. So now we need to put one in here. So what we don't want to do is we don't want to overwhelm our audience. Well, let's put one up here first. Let's get a little bit of stuff in this well here. Need some darkness here. I don't understand what I'm doing wrong here.
Well, you're falsifying data.
Well, is that wrong? Now I'm going to make it wider.
Jerry, I found sometimes the interactions between programs sometimes get you problems. Sometimes it works, sometimes it doesn't. It's just interactions.
Interactions among devices and drivers.
Well, we're going to do it. We'll get there. Be patient. Oops. It's hard to do.
Are we going to be co-authors?
Of course. You'll see. I've already made provision for that. There's a little.
Thanks, but I'll keep my name off of this one.
You're not senior enough to be forcing your way in as a co-author.
Okay, now let's make a lot lighter here and a little gamma here. Or no gamma, low gamma. I don't know why I'm not getting it. I am getting it, just so light. Okay, now we want our Drosophila. So let's give it a little more opacity. Let's see what we get, a little more width. Like that. Get just a little bit more width. And then let's hit it with get the gamma. Move this down so our edge will be a little rough. I'm doing a dot, dot, dot. You can't see what I'm doing, but I'm doing dot, dot, dot to fill it in slowly. Okay, so now we've got all our red lines done. So let's just make our red lines disappear. Okay, so now we'll still get caught because somebody's going to find some pixels. So what we want to do is we want to go to another layer here, and I'm going to make these other layers disappear for a moment. And I want to show you how to create a mask. We want to create random noise. Now this will be too dark, but I want to show you what you do. This is a filter mask. And it creates all of this quote random dots. So that if there are pixels underneath it, a pixel pattern underneath it will be lost with the random dots. Now this would wipe out the picture, of course. So what we're going to do is we're going to do this again, but with a noise, instead of opacity of 99, let's make the opacity 4. And I don't know what the level is, but it does something. And we create a filter mask. Auto saving, document is busy. What does that mean? Let me do that again. Make that 4 and draw the level down. Now, can you see this on your screen, all these very, very faint dots?
No, they're not really coming across well.
Well, let me do it again at
Not really. And also I have a more basic question. This is a great demo of graphing software, but I'm losing the bubble.
You're losing the bubble.
Yes. Where are we going with it?
You'll see. Can you see this mask here, the tiny grays?
No,
Well, I'll make it just a little bit darker for you, and then I'll make it the way I want it. See, I'm going to make it lighter. I'm just showing you how to make it lighter. This is. It's updated. Can you see those tiny dots? You can't see
No,
them.
not really getting that.
Okay. Well, I don't know why you can't see them, but there are tiny little dots on this page now. And the tiny little dots will change the underlying pixels in a random way so that when you show the other layers, if you got this random noise, the pixels will be changed so that you won't be able to find a pattern to it. So now we have to take this graphic and we had to snip it out of here. Put it in memory. And let's put this down. Now we need some paperwork. So you asked if you would be co-authors. As a matter of fact, you all are. This is a journal page I'm setting up. This is the Journal of Nefarious Skullduggery. And we're going to show the transfer of Drosophila fruit fly gene XA2042, I made that up, into a human lymphocyte stem cell lineage. And the authors are me and all the other gonnists in the Lexington Computer and Technology Group. Okay, so now let's just look at the layout of the page. Here's a section break here. So we're going to make this, everything underneath this, we're going to make this 2 columns to look good, 2 columns. Now we're going to insert some dummy text just so we can see how the setup looks. Now, is anybody familiar with ipsum lorem? Ipsum lorem was invented about 350 years ago by an unknown printer. And let me press the return. What it's used for is it's used as dummy text when you're setting up. I just want to use the Georgia font. When you're setting up a page, it permits you to set it up before you receive the author's text, so you can see what it will look like, and and you can show your customer what it's going to look like, so they can choose what they want, how they want the setup to be. Now, lorem ipsum comes from the expression dolorem ipsum. It's from, I had 5 years of Latin, so this means something to me, maybe others too. It comes from Cicero's book, De Finibus Bonorum et Malorum, on the limits of good and evil, the boundaries of good and evil. And most of these are snippets of sections, sentences from that. But I've also seen some of them from his other book, De Naturam Deorum, which is on the nature of the gods. And we're going to add, let's just, for your approval, let's say we're going to put our data in here. Now, the best way to do it is to insert a table format, because if you have a graphic and a legend, you want them to stick together if you move them around. So it's better to have a graphic and then the figure, the legend will be here. We usually want the legend to be in a sans serif. So we'll say, let's pick Calibri. It's a good sans serif. Okay, now we're going to put in our graphic. Now it's a little too big, so let's move it to the front so we can work with it and knock it down in size. Now, when it gets knocked into size here, it sticks into this, it'll stick in our pro, so we can move it all around. Now we need a legends here, so go. We need immunoelectrophoresis. And now let's label these columns. So we'll insert a text box. We're going to put it up in here. So we can label our columns. Oops. I'll move it down just a tad so I have some room for this. I'm going to move it in front. Okay, now we want to work on the fill here. So where's we don't want any fill. We don't want any outline. So now move this up just a tad. Keep it on the graphic though. Okay. So here's column A, column B, column C, column D. Now, immunoelectrophoresis. So column A is control, and you didn't do anything. B will be normal. Site DNA. C will be, oops. C will be DNA with, I'm sorry, stem cell descendant with and D will be sham insertion. Now, insertion. Now we want to insertion. We want to tell them that we stained it because otherwise you wouldn't be able to see it. So now we want to show how smart we are. This is the best stain for DNA, but it's a carcinogen. So it has to be handled carefully, has to be disposed of carefully. So only experts would be using this. And it shows that we are experts. And of course, we got a little condescending, but we're going to put agarose gel. Now, to distract the eye, you want to have a shape to distract the eye. So what we're going to do is we're going to put in an arrow at the Drosophila DNA. So let's put red arrow, and oops, we just need a little more space on this. Let's draw this down a bit. We want to align this left. Now, the arrow, blue isn't any good. We want to have a distracting arrow. So, there we go. So let's go here. And let's give it some width. We're going to give it 2-point width. And let's make it a bright red. So that ends up very distracting to the eye because you want people to look at the arrow, not the diagram. Okay. So now we have make it a little smaller so you can see the whole page. And now we don't want borders on this. In the print business, a line is called a rule. And you don't want them showing. The printer's expression is, the rule is as few rules as possible. So I made the lines transparent so it appears like it's floating. That's a better way to do it. So now we've falsified our data, and I did this in 5 minutes without my graphics pen working. Give me 2 hours, and I can make it. You just won't even suspect that there's falsification. But even if you look at the pixels, my mask will hide the pixel pattern, and we're good to go. So that's how you falsify data. So let me go back. Now there's other things. Now this is what it would look like. This is another one I did. This is what it would look like in our publication. Now there's one more point I want to show you. And that's about file size. I drew this blossom on my graphics pad is the Chinese brush style where it goes from fades from one color to another. And if you look at the critter, like it's a scalable vector graphics file. It uses the suffix KRA, but I'm sure it's just an SVG file. But look at the size of that. You will never want to let this out of your hands. This is if you have a graphic that you're showing, you never want to show that it's this big. They'll know that you fooled around with it. So you could export it as a PNG file, which has a little metadata on it. But if you take a screenshot or a JPEG, it has almost no metadata. That's far safer. Now you're going to submit it to the journal as a PDF, but somebody's going to ask you for the Word file somewhere along the line. So you got to use these. You could put a bitmap. It has a big file size, but I think that's just because every pixel is specified. I don't think there's any real metadata on that. And then to where suspenders on the belt, you go to Windows, and you look at the folder and you remove properties and personal information. So there's a dearth of stuff on the file. So now we've gotten where we want to go. So in summary, data fabrication, the advantages are enormous time savings, enormous. We did a year and a half's work in 10 minutes and 3 hours. I'd be another week to write it up. I'd be great. Reduce stress of laboratory work. We already know we're going to end up with a nice clean experiment, gives clear definitive results, and it does add, I admit, value to our resume for academic advancement. I admit that. The disadvantage, of course, if you get caught, you are totally screwed. And that is my talk on scientific misconduct.
Hooray, hooray. Very nice. Now, how is it you know all this so well? Is this known technique, or are you actually
No.
polishing?
No, I have created this.
So you can now write the handbook.
I did write the handbook.
I see that.
On fabrication.
Right.
It probably would sell quite well. And when it comes down to it, it'd probably sell.
And Jerry, you can email it to us so we can print it to our scanners.
You're right.
Yeah, well, we've at least got our cover page, our face page.
Yeah. You're in good shape. And that it just takes some practice, but the amount of time is it's tremendous time saving. And I like pictorial results. Pictorial results are always better than just stated data, because who knows what state of data is,
Well, you should license the Chat GPT, and then the combination what you've got, you have the ability to crank out papers at a phenomenal rate, and you just sit back.
That would be a dead giveaway. That would be a dead giveaway. You got to remember that's a year and a half's worth of work. So you got to have, now you can invent co-workers that can help, but you don't want to publish a paper every week. They'll know you're cheating. You got to remember not to be a gonoph. Don't leave a trail. Everything you do, you have to look at it and say, how are they going to catch me?
So what about?
Well, that's why you use people who are in remote parts of the world. That's the old trick that the newspapers always get. Quotes from somebody or other in Azerbaijan or something like that, and nobody can track the person down who said it. So
now I would, however, pass on my father's advice. He was a lawyer, and he said, whenever you commit a crime, you never have a partner, and you never tell your wife. You never tell anybody. That's essential in committing a crime. There's no partner. So if you want to do this stuff properly, you don't have any help at all. You just do it yourself.
Classify it.
Right. And then put it in your garage next to your Corvette.
Yeah,
But
the problem is, in most science nowadays, there isn't just one person. There's a whole
You have to
host
make
of
up
people
people
involved.
You'll have to create people.
But that becomes a visible trail. Where they come from?
It does. What I would do is personally pick an experiment that you can do like StayPal, experiment that's very simple that you could reasonably do by yourself. That's how I would do it.
Do we now have a journal of falsified results?
Journal of Nefarious Skullduggery.
Yeah. Well, I don't know. It seems to me that's a little too sweeping.
It's a little too on the nose.
Journal of non-reproducible results. It's
falsified.
Then you get yourself an ignoble prize.
You're right.
Yeah.
So,
I think actually getting a few other people involved, even if they're fake people, would make it more plausible in the scientific community.
Unless somebody tries to track them, see who they are. You know,
Then get some real people.
everybody's on LinkedIn and everything. It's a tricky business, creative person.
Yeah, then get some real people and make it worth their while.
No, they become your partner. All of a sudden, they're your partner. You
Well,
don't.
Dead people.
Yeah.
For a few of them, you can have them be memorials to your tragically
No,
dead
yeah, right.
partner.
Tragically died would be good. That would be good.
Yeah, you could do that. Yeah. The bus ran off a mountain road in Azerbaijan,
and that's who you bring
all
in,
my
yeah.
coworkers were killed.
Yeah.
Okay, it's getting to be 11:30. Do we have more that we want to cover that's of substance here?
Yeah, let me ask Jerry a question here. When you read a paper, how do you go about finding out whether it's fabricated or not? The data.
Well, what you do is you look for irregularities, things that just don't seem right on basic science, basic biology. You look for things that it's like you look at the Sputnik V and there was no descent in efficacy with age. That's almost unheard of. So you already say, how could that be? Because you look at other data from adenovirus immunizations, that isn't so. So you look at things that the author would want you to have that just don't seem right. That's why you have a slightly distorted truth so that they seem right.
Hey, Jerry.
Yeah.
Are these refereed publications?
The falsified ones
Yeah.
that I just did?
Well, no. I mean, let's say the Sputnik ones, for example.
Sure, there's plenty of criticism of that.
No. Did it have to go through referees first?
Yeah.
Well, that tells you something.
They
about
requested
the
They requested the data, and Lancet
tells you
published.
something.
Lancet went on to publish because they wanted to be first. Lancet went on to publish despite the fact that the referees had asked for the primary data.
Okay.
They took that. Lancet has gone out on a limb many times, and they've been first and great many times, and are renowned for that. But they've gone out on a limb, like with the Psi, they've gone out on a limb without primary data, and they've gotten screwed because of it.
So there's an incentive to publish without adequately fact checking.
Yes, to be first.
Yes. That
'Cause if you
also
have to give that to those referees, they'll be 6 months before they get around to it.
that also happens in spot journalism.
What's spot journalism?
That means where you're doing a breaking news story.
Oh,
And
right.
you
They
have
don't
to
check
be,
their facts, right?
you have
They
to
don't-
be first, and sometimes they will use raw footage or raw data. We
Yeah.
have two hands up. Let's do Carl first.
Yes, I wanted to, Jerry, although you showed a really sophisticated way to fake an image, it strikes me that that there might be software available that would look at an image and see if there are any pixel patterns that are replicated within the image as a way.
Right. That's why my mask would prevent pixel patterns from
Yeah.
being recognized.
But there
'Cause
is such software.
No, because it's really computer's not really random, but it's pseudo random that there's no pattern to pick up.
Yeah, that usually works, although sometimes the mask is too perfect.
Well, that I can't tell. I'd have to have a computer authority to tell me that.
It depends on the algorithm that's trying to detect it.
Yeah.
And Ted had a question or a comment.
Yeah, so my question would be, I mean, obviously these imagery things, that's a sort of an artistic skill, but the other kind of thing is where you have a bunch of data, these particle accelerator experiments and things like that are a classic example, and then you have to filter the data, and you always have to filter the data because there's always things that end up in your data that are not part of what you're looking for. But you can build a filter in such a way that it finds what you're looking for, whether it's there or not. Do you have any comments about that kind of a situation?
No, well, I don't know enough about that kind of data filtration because I don't manipulate. I used to see that when they had the original cyclotron, they used to show the stuff shooting across the on a piece of paper. I don't know how they filter that.
Yeah, well, I mean, the old days, they had graduate students that, they gave them these rolls of exposed film, and the graduate students had to go frame by frame through these things. Now you pour the data through a signal processing system, and the computer searches billions of events and looks for this one thing that gets you a Nobel Prize.
And filters can be tuned.
Yeah, but
absolutely.
don't you
Yeah.
do, I mean, like in LIGO where they, you had people taking, different groups taking the same set of data and extracting out the signal. And then only after they had done it did you check across the groups whether they were getting the same results. So you would have had to have collaboration. If you were going to do fakery, you'd have to have separate groups collaborating.
Well, in some cases you can't. I mean, you have only one
That's
set
true.
of data, and the data gets processed before it gets archived, because when you have billions of these events and stuff like that, you can't store terabytes all the time. You store megabytes and or maybe gigabytes, but you don't store terabytes or petabytes. And so
No,
I think
ultimately-
it's getting harder and harder to make sure that you're not seeing artifacts introduced, not necessarily always by intent to be deceptive, but just sometimes deception ends up built into it through the way the signal processing is constructed.
Wasn't that how Joe Weber got stung? I mean, he thought he had discovered gravitational radiation.
course.
And it
Well, that
turned
was
out to
quite-
be something in the software.
Okay,
Yeah, well, it was noise on the transmission lines.
we have
And-
Joe
something.
Weber wasn't falsifying.
No, he didn't falsify it on purpose. It was false, but
Yeah.
Same thing was true with the so-called neutrinos moving faster than the speed of light.
Right.
Well, there was a Chinese group that reported that they received a signal from outer space, a microwave signal from outer space, but they only received it when they were heating up coffee in their kitchenette.
Yeah. Well,
So
that's
much-
that's why Penzias and Wilson swept out the horn there in Homedale for pigeon droppings.
We have a question from chat, and that is, do we have an estimate of what percentage of papers are falsified and what fields might be the greatest offenders?
I read about 15%.
Mm.
See, but some of them don't influence the conclusion so much, but they're just massaged a little bit.
So 15% might be massaged a bit.
I mean, when I did research on dogs, we had to you do a dog experiment, it doesn't come out particularly good. You just throw it away.
Okay,
You didn't put it in your data. Now we said, oh, it just didn't come out right. And it never appears. Well, it's like Mendel if you have a plant that isn't quite clear, you put it in the category that you want to.
yeah. Any idea what kinds of fields would be more prone to this sort of thing?
I would know that.
Follow the money.
Yeah, right. Yes, that's true. That's it. That's it. That's exactly it.
Yeah.
But academia is also just academia. Now, China, it's really publish or perish. And there's a lot of falsified Chinese data that gets published. That I know. They had one paper showed how they could do chest surgery under acupuncture. And they show the chest open. Well, you open the chest, the person can't, and the person was not on a ventilator. The person was just lying there smiling. Well, you can't open the chest. You can't breathe. The person couldn't be lying there smiling, but they didn't understand the physiology.
Well, unless they were already dead, and then they could
Unless
be
he was smiling. It was
Yeah.
a Risus sardonicus. He was smiling.
smiling.
Okay, anything else important? Anything else important we want to get to?
Well, I think we should do something about Chat GPT. We've been talking about it,
I can't get
informally.
on it, Ted. They say it's full.
I know. I've been having trouble getting on it too. But I keep seeing stories coming out from the media about it almost on a daily basis now. So
I think I can take the honor of the first lecture, at least since I've been in the club, that has no redeeming value.
Don't
Okay.
overdo yourself.
Don't let it go to your
I deserve
head.
an award for that.
I think this does have redeeming value because it gives people a new sense of way of being skeptical about what you see sometimes.
So Ted, do you remember who it was? What was it? The solid state guy? Was he an Austrian, Swiss? Who a few years back? I mean, it was a terrific piece of fakery in physics.
Yes.
Was it?
Okay, before we conclude, is next week, what I'm looking at in the schedule is that March 1st is listed as the planning meeting. So is John Rudy still on the call? Okay, we're going to have to figure out exactly how we're notifying people if there is in fact a planning meeting. I seem to be the only one in the planning committee who's on the call still. So I'm not sure exactly whether or not we're doing that next week.
I think you just want to tell everybody to show up ready to plan. Come with suggestions for the
Yeah,
next
we will need an email
next
about
morning.
that
Yeah.
to be
Good.
announced. So I'm going to make a note in my notes, and if we want to continue the discussion, I think it should get concluded soon. So
Fine talk, Jerry. Thank you very much.
Yeah.
Thank you. Thank
thanks,
you.
Jerry.
It was entertaining and as I said, somewhat informative too, because you see so much of this stuff, and especially if you're not really deeply embedded in the field in question, you read the paper and you go, gee, that doesn't sound right, but I can't put my finger on it. Now I can start to put my finger on some things that seem
So in the field, I mean, artificial or synthetic data has been around for a long time. Usually the domain is textual. Now you're getting into, graphics and such. And there are techniques that image processing does, like two-dimensional FFTs or FFT processing that sometimes brings out the artifacts that show the chain of custody or even how the image was scanned in, even if you try to anonymize it to a certain extent. So there are experts out there that you can tell what device was used to for example, to take that picture, because it was all digitally produced. There was no underlying camera that should have been there.
Right, that's getting it right. Just to strip off the metadata as best you can.
Yeah, there is this cat and mouse game between the more sophisticated detection schemes and the more sophisticated ways of faking things. And I think maybe that's about as far as we can go with that.